For cells to do their jobs, they often need to move. For example, when embryos develop, lots of the complexity they gain comes from cells migrating to new sites, to build new structures and to change their organisation. And if you get an infection, your immune cells need to migrate from your blood vessels towards wherever the problem is. So it's absolutely vital to life that cells do this right. Without accurate movement, embryos can't develop correctly, and infections can't be neutralized. Sometimes it goes wrong. Cancers spread, for example, when cells migrate out of the tumour and into the rest of the body. My lab studies the signals that steer cells. We're particularly interested in chemical signals that diffuse through the tissues, making gradients, and the gradients tell cells where they should go. This is called chemotaxis. Now, there are quite a number of labs studying chemotaxis, but we have a particular expertise, which is cells that make their own gradients. If someone else makes the signal, your cell's job is simple. It's just to respond to the gradient. But we study cells that actively break down all the attractive molecules, so they generate their own gradient wherever they are, low where the cells are and high outside. The gradients are much more dynamic and much more interesting because the cells can read lots of information about their environment. For example, if there's a short path and a long path towards a new source of attractive molecules, cells can tell the difference by reading the steepness of the gradient. We recently showed that cells can use this information to find the best way through a maze. Now, this is fascinating, but it's also a very good model for what those same cells need to do in a developing embryo. In this project, we're going to study a different thing. Many of the attractive chemicals we're interested in stick very strongly to the extracellular matrix, which sticks cells together in tissues. This makes the attractants diffuse differently, much more slowly, and this slower diffusion changes the gradients. It makes them shallower, and it makes them change more slowly. So in this project, we're going to find out whether cells can learn about their environment by reading these gradients. Do they prefer a path with one kind of matrix over another? Can they tell the difference between different sites, between breaking down an attractant and seeing how this forms a gradient? We'll use a wide range of techniques. As well as lots of microscopy and cell-based assays, we'll do biophysical tests to see how different matrices change diffusion. We'll do biochemistry to see how attractants are broken down. And we'll use computational and mathematical models to see what we think the cells will perceive. This means that the student who does this project will get a really broad training in pretty much the whole of modern biology. We'll collaborate with a company called Manchester BIOGEL, which makes hydrogels for cell to migrate on that change diffusion in different ways. And we'll work with academics at the Universities of Glasgow and of Birmingham. Combined with all the interesting opportunities that the lifetime CDT can offer, this should be a progressive and fun way to do a PhD.